To begin by thanking Gene and the organizers for this opportunity. I have been here for a very short time. So what I'll tell you today is an introduction to the problem that we are interested in and how studying this from a different angle is getting us closer to solving this than what has been attempted for more than, say, about 100 years on and off in biology. Okay? So the problem that I'm interested in is, do environmental effects that happen in the duration of one lifetime in an organism have consequences for the next generation? This is a problem that has been contemplated in biology on and off for more than 100 years. It has had a checkered past. So I'll tell you, up here are some of the citations that are worth reading in this. And there have been effects that have been observed in humans, and by the nature of this study, it has to be correlative. And some of the things that have been observed are grandparental diet can have consequences to the health of grandchildren, and early parental smoking can have consequence to the health of the son. So this is paternal. Okay. Now, of course, in humans, you can only do this sort of retroactive correlative studies, but it is possible to do causal studies in rodents, and that has been done in rats and mice. I've highlighted here some of the recent good work that's come out of this field. And they include paternal diet that leads to health consequences in daughters or sons and daughters. And specifically, the famous endocrine disruptor vinclozolin can lead to sperm defects that last many generations in rats. So these observations raise the problem, how can this happen, right? Because it's unimagined as to what the system could be that's propagating this information forward. And the reason is, for you to get any information from the body cells that are experiencing the environment to the next generation, you need to go through the bottleneck of the germline. Okay, so if you imagine any organism, there is the germline and it has somatic cells. Generation after generation, you have this divide, but the germline is continuous. The soma waits for the germline to give rise to it. Right? If an environmental insult, say, perturbs information in the germline, that could then be passed on. But if it perturbs something in the soma, there needs to be first transport of that information into the germ cells for subsequent propagation. Okay? So this is where the problem is for the last 100 years, because our understanding of biology, any biology, is incomplete. Okay? So you can have people who would do correlative studies where there is starvation, let's say, and then subsequent effect later on. You have no way of knowing what did starvation do to the body, and what was that transduced into, which was then transferred to the next generation, right? So what you really need is a straightforward way of going after one particular gene and following it generation after generation, okay? So it's exactly this that Andy Fire and Craig Mello stumbled upon, sort of sideways, when they did this odd experiment. When they added double-stranded RNA against a gene by injection, let's say, into this 1 millimeter long worm. I'll introduce you more about it, called C. elegans. What they saw was that there was silencing of that gene remarkably not only in the cell where they injected the double-stranded RNA, but throughout the animal and in subsequent generations. So this finding is startling because all you need to remove a disease gene is double-stranded RNA that matches that gene. And this medical importance was recognized quickly, and they were awarded the Nobel Prize in 2006. And then, but I think there's something more here in this experiment. It is telling you that in an animal, it is possible to get sequence-specific information throughout this animal and across generations. So this is the tractable model that I was in search of when I finished my PhD and said, what do I start doing my postdoc on? Okay? So with this tractable model then, now we can at least figure out what is the road or what is the pathway through which information can be transferred. okay? And thereby get at the mechanism of such transfer mechanisms that might exist, okay? So this is not a process that is just a curiosity of you adding double-stranded RNA from the outside. It turns out that more than half of your genes in many animals are under the control of small RNAs. In C. elegans, the worm that I just told you, there has been extensive studies, and this has also translated into extensive studies in many other organisms as well. So in the decades since its discovery, this is sort of the skeletal pathway of RNA interference that's come about. You have long double-stranded RNA that is processed by a conserved enzyme that you and I have called dicer to give you these short double-stranded RNAs, of which one of those strands is removed by another enzyme which you and I have as well called argonaute, which will then find a matching mRNA and silence it. Now, the real explosion in understanding the role of RNA came from simply sequencing a whole bunch of these small RNAs and seeing where do they come from. And when that experiment was done, they found that it matches more than half the genes in the genome. And you know that that's got to be an underestimate because the way the experiment was done, you just took all the RNA and sequenced it. If anything was low abundance, you will miss it, okay? And not only that, even if there are 1 to 2 base pair mismatches, you can get silencing, okay? So this is a rather ubiquitous process. So where does that leave us with this idea of RNA moving about between cells? Now this happens in plants as well, and in plants it has been easier to imagine what's going on because plant cells are connected by large intercellular bridges called plasmodesmata, and they have vascular tissue much like us called phloem. And recently, a couple of years ago, it was shown that these small RNAs can move between cells in plants and long distances in plants, okay? And more remarkably, these silencing small RNAs that move between cells can lead to epigenetic modification. Now, this is a word that's often used but not very well understood. What is meant here is anything that's not a genetic change, okay? So in this specific case, it was DNA methylation, not a base sequence change. Okay. So this is possible in plants. How about in animals? Animals don't have plasmodesmata that connect other cells, and I'll tell you in detail about work that was done in C. elegans. So let me quickly introduce you to a little bit of C. elegans anatomy. The worm is about 1 millimeter long, and I've highlighted only some of the key features here. That's a cutaway view on the right. Okay. It eats its food, bacteria in the lab, through its throat, and then you have a gut lumen where the food enters, from which it has to be absorbed by the gut, and the nutrients can be dispersed from there. The reproductive tissue is also found inside in that inner tube, and on the outer tube, just surrounding the cavity where there is fluid, is the other tissues, which include muscles, skin, and neurons, okay? A dorsal and ventral nerve cord. Now this is a broad stroke anatomy, right? We're not going to go into the details of C. elegans anatomy, but this is important to keep in mind for the things that I'm about to tell you. So how do you look for genes or proteins that are required just to transport this signal in the absence of being able to, when the only readout that you've got is silencing, right? So a screen was set up in Craig Hunter's group where they fed this animal bacteria that expresses double-stranded RNA. Remarkably, the gene that matches the double-stranded RNA will be silenced in the worm, and also express double-stranded RNA from this throat. And then the matching gene, in this case green fluorescent protein or GFP, was expressed in the body wall muscles. And they simply look for worms that look like this, where the pharynx or the throat is silenced, but the body wall muscles are not. And these would be specifically defective in the transport of RNA between cells. Okay? So what they found was that most of the mutants that came out, there were 200 mutants that came out of this, and 100 of them were SID1 and SID2 and SID3. Okay, SID2 and SID3 were the remaining. Now, I'm going to summarize about a decade's worth of work in this one slide so that I can tell you unpublished work from my young lab of 1 year in the remaining time. Okay, so what we've now gathered is that double-stranded RNA that's expressed in any tissue can cause silencing in other tissues by making mobile RNAs, which are forms of double-stranded RNA, and they enter the other tissues through a conserved protein called SID1. This literally is a transmembrane protein that sits on the membrane and can let double-stranded RNA right through. This is also conserved, and there is endocytosis that occurs, which is promoted by a conserved tyrosine kinase. That's what SID3 ended up being. Okay? There are homologs of both these proteins in us, but the screen did not reveal any of the proteins that are required for making it and exporting the RNA out, nor do we have any understanding of what an exporter cell is in an animal and what an importer cell is in an animal. Okay? But as I told you, the idea of using double-stranded RNA to shut off any gene at will is of great medical importance. And one of the companies that's pushing this forward a lot is Pharmaceuticals, and they, along with Marcus Stoffel's group, thought up this experiment where they wanted to reduce the levels of apolipo-B, which is a known molecule that's involved in hypercholesterolemia, but there are no small molecule drugs, so ideal candidate for this. What they did was they coupled the small RNA that they were going to try to use to silence apolipo-B with cholesterol indicated by L there. The idea being it'll be bound by LDL, delivered onto liver cells, and then they'll get the siRNA in to silence the matching gene. But they found something remarkable. The delivery worked, but the siRNA was internalized without the LDL being internalized. And not only that, if they used antibodies that blocked SID1, the mammalian SID1 this time, the siRNA delivery was prevented. Okay? So there seems to be a conserved way for RNA, double-stranded RNA, to be taken up into cells across many animals. Okay? So what would the worm use this for, or what is the advantage that it might gain from using this? So an early idea that came from looking at repetitive DNA sequences. That's 50% of our genome is repetitive DNA sequence, and we don't understand what that's for. And through a lot of work in Palm Beach East, it's now known that heterochromatic DNA or repeat DNA can be reinforced by this feed-forward loop of small RNAs that are made. Okay? So in the work, C. elegans, when you try to express green fluorescent protein, you usually make many copies of it and you integrate it into the genome. For example, shown here is expression of GFP everywhere. Okay? Now, if you remove a protein called ERI1, which again is conserved with us as well as in C. elegans, its role happens to be to chew up the ends of these RNAs, then you can get really enhanced silencing in its absence. Okay? So when you get enhanced silencing in its absence, it's pretty widespread. But look what happens if you then block the transport of RNA between cells by removing SID1. You still get some level of silencing in some cells, but not in all cells. Okay? So it seems like there are two kinds of cells in an animal. Those that can efficiently silence double-stranded RNA or DNA or repeated DNA, and those that cannot, okay? Even though they are of identical genotype. And what seems to be happening is that there is a transport of RNA between cells that then allows the silencing in the second cell. So Haile in the lab, he's a very talented undergraduate. He decided to push this and figure out, okay, why is this happening and what are these two kinds of cells? And C. elegans is really suited for this because we know the name and position and lineage of every single cell, okay? Every single somatic cell. And the gut lineage is particularly suited for this because all gut cells come from one cell in C. elegans, and they get arrayed like this, okay? So what Hai did was he took a specific stage of the animal and then simply measured the amount of fluorescence in every single gut nucleus and related it to the lineage. Okay? And you can see there's a pretty dramatic difference, up to 100-fold, that's possible in this genetically identical adjacent cells. Okay? And if you did this, or if I did this for 60 different worms and arrayed them across, we can then ask the question, is there any one cell that is always specialized as the one that makes a lot of RNA silences and is therefore the exporter cell? And if you look down here, you can see there's no single cell that does that. Okay? So there seems to be that with the same genotype, the animal's making a mosaic inside its gut. And not only that, Hai then did a time course looking at the same worm starting early on in development and watching it all the way through. And the remarkable finding was that the same cells stay on or off throughout development. What that tells you is that there is a fate decision that seems to be happening early in embryonic development, which decides whether you're an exporter cell or an importer cell, and Hai is pushing this further. And more broadly speaking, in general, it is not clear how random events during development influence our health as we grow, and this can serve as an easy, tractable model to understand such developmental events. Okay. So what about the genes that are involved in in exporting these RNAs? I told you about one pioneering screen that was done in Craig Hunter's lab where I was a postdoc, but in that screen, they added double-stranded RNA in the bacteria which they fed the worm, and that was needed to see efficient silencing. Okay? So you can imagine if there was any gene that's involved in exporting RNA, it would be masked by the fact that you have a second source of RNA that's coming from the food. Okay? So we now know that empirically, neurons are really good at exporting double-stranded RNA or a signal derived from it, and it can silence genes throughout the animal. Okay, so in wild type, if you express double-stranded RNA against GFP only in the neurons, then you get silencing of GFP in magenta here everywhere. If you had a transport mutant, the GFP comes back on. Okay. So this then is a powerful way for us to identify many mutants, and we identified a bunch of mutants in a pilot screen, and then two graduate students in my lab, Snusha and Sindhuja, decided we'll just do whole genome sequencing and figure out if we are getting things that we want. I wish this technique existed so robustly when I was doing a PhD. It would save me 3 years at least. Okay? But it works remarkably well. So what we can do is we can just take all 19 mutants and simply do whole genome sequencing and identify all the mutations in them. And with a little bioinformatics, you can get to the gene. So here are a couple of mutants whose genomes are kind of schematized here. The blue and yellow boxes are genes, and the other things are mutations. If you remove all things that are filter off non-genic mutations, you're left with, say, this set. You can then filter off silent mutations, and then you would expect some mutations to be there, which are this background mutations, which are never induced by your mutagenesis. You can remove that, and then you're left with a fewer set. So how few? If you look at the number of mutations that's there in all 19 of them, it's under 50 on average, which is remarkable as a way to proceed with the screen. And not only that, if you're really lucky, you might actually find the gene right away, because you'll have 2 or 3 different mutants that have mutations in different places on the same gene. And indeed, Sindhuja and Snusha were lucky, and we found our first gene, I think, simply by staring at a computer where you could see that there are 3 different mutations in 3 different alleles. And they're pushing this now further to saturation, because of the 19, only 6 were mutated in genes that were previously implicated in RNA silencing. So now this is a powerful way to just identify all the genes that you want. So we think there are going to be specific conserved genes that are involved in making and transporting RNA out of cells. So the perspective that you get from thinking about this conserved mechanism is perhaps this is a kind of signaling mechanism that's similar to steroid hormone signaling, because steroid hormones can cross membranes because they are lipophilic. Then they usually bind a protein that can use its sequence-specific DNA binding ability to have downstream effects. So both these features are present in this system, where you can have double-stranded RNA that crosses the membrane by virtue of SID1, this conserved protein, and it can cause sequence-specific effects because the sequence information is on the RNA. The one difference is that the sequence-specific effect is actually to the same sequence and not different sequences as shown here. Same sequence as the RNA. Okay. So where does that leave us in thinking about the problem that I introduced you in the very beginning? To be able to analyze what is the information that is transmitted across generations, people have often looked at what is the information that persists. For example, is there DNA methylation that stays across generations? Is there chromatin modification that stays across generations, right? So that suggests that it could be either causal or just correlative, that it just happens to be there, okay? So a clue as to whether RNA could be the one that's transmitting the information came from this experiment published last year. So in worms, because we have this easy way of silencing any gene you want simply by feeding bacteria that have that double-stranded RNA sequence, you can feed the worm double-stranded RNA against a gene called DUMP11 that makes the worms fat, called DUMPy, and that can last in the second generation and then goes away. Now, how long an effect lasts is also gene dependent and condition dependent. We don't yet understand what that is, but in this case, there was a very interesting follow-up experiment, which was to then look at the parental generation, the first generation and the second generation after, at small RNAs, as well as at a repressive chromatin mark called histone H3 K9 trimethylation. And what was found was that while in the parental generation there was a lot of small RNA, there was very little H3K9 trimethylation. And in the second generation, H3K9 trimethylation accumulated. So it's a real possibility that RNA is what is transferring the information across generations. So this crystallizes the focus in our lab to a very specific question, which is what exactly carries this sequence-specific information from body cells to germ cells? So we have been able to reduce this amorphous problem of what environmental effects can go to the next generation to this very specific question. And Ed Traver in the lab is systematically pushing this to figure out which of these. Are these RNAs that are early on, or are they RNAs that are produced after the chromatin mark? All right, so, oops. Okay. So, in conclusion, I want to tell you about the way in which we are thinking about this problem. So for RNA to transmit information across generations, we have to think about three things. One is germline axis, because how is the animal made? Can the animal, the somatic tissues of the animal talk to the germline and transfer information to the germline? The other is, even if there's one somatic tissue which can do that, is that exposed to the environment, and can that trigger the transport of RNA? And finally, as you saw, even within that tissue, chance dictates whether that cell is an exporter cell or not. So in short, it depends on nature, nurture, and chance. And with that, I'd like to mention the people who have pushed this program really fast over the last 1 year. Hai, who's an undergraduate in the lab, who's doing the differences between exporter and importer cells. Sindhuja and Snusha are graduate students who are set to figure out all the send genes. Ed is pushing the specific RNAs that may be transported from somatic cells to germ cells. Soraya is looking in detail at the transport between tissues within the same animal. And Linnell is helping us go after the endogenous RNAs that are from the native genome that could be moving about in this way. So thank you for your attention, and I'm happy to take any questions. Okay, yeah?
Is there any evidence for this, or is there any clue as to what the structure is of the import protein that brings the double-stranded RNA?
Right, so the only thing that has been done thus far is it's a transmembrane protein. It has 11 transmembrane domains or so. The only thing that's been done so far is they took the extracellular domain and did an NMR structure. It looks like it's a tetramer, which forms a hole in the middle that fits the diameter of a double-stranded RNA.
And that's in c.elegans, right?
The CID1 is a conserved protein. The extracellular domain that they used was the C. elegans extracellular domain.
And was it homologous to any eukaryotic or any human or any higher order species?
There is a CID1 homologue you can fish out by blast from humans. So, yeah.
Actually, I have two questions. First,
Sure.
can you repeat what the acronym SEND stands for?
Oh, silencing exported from neurons defective. It's contrived. It just means it's RNA mutants that are defective in transporting RNA from neurons to any other place. Yeah.
And also, so have there been any studies done with other animals besides C. elegans?
Right.
Is that something that's being...
Right. So in the interest of time, I didn't push it too much into the other organisms, but there are a lot of other organisms that people have studied this on. I think one of the best studies was in Drosophila, where they found that aging can lead to the accumulation of transposon silencing defects, and that is transgenerationally inherited. Yes?
I've read that there has been some evidence whereby a parent, before they have a child, if they had been exposed to some environmental insult that could lead to cancer, but that they fight off that environmental insult and don't develop cancer, they're yet to be conceived, and future children might have lesser chance of developing that type of cancer.
Right.
It's sort of an environmental induced later,
Right.
partial immunity.
Right.
How could that be explained in this system?
So there have been many studies of that nature, and they're often on cancer or obesity, diabetes, these complex diseases, cardiovascular disease, etc. My take on that is right now those studies are correlative and they're at the level of phenomenology. We can't get to the mechanism, and it's largely because we don't understand any of those words really well. Okay? So we just have to stick with what we can clearly understand, and to me that is, if you can say this gene is what's getting silenced across generations, that can be followed, right? So there have been many publications like that, and I would say that, you know, jury's out on if that stands the test of time. Yeah?
You mentioned earlier in your talk that this debate of exporter and importer cells are sort of, see, what happens if, let's hypothetically suggest that the mutation appears de novo, but can a mutation appear in exporter cells and the mutation be in the protein or process that is a result that affect as a result of mutation be silenced through something like by importer cells versus exporter cells?
Okay, so if I understand what you're getting at is, if you're making a mosaic model of your any tissue, such that one...
What I'm trying to get at is this importer-exporter phenomenon a basis for some mutations appearing as a mosaic phenotypically?
Sure. It is very well possible that if you get a somatic mutation, then it depends on which cell you got the mutation in for a phenotypic consequence. Yeah. And one of the interesting things is transposons, which are remarkably well silenced in the germline, are actually not very well silenced in somatic tissues, and they're hopping about all the time.